Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. So welcome to my lecture. So this is lecture 2 of your excretory substances and their elimination. So this is a part of human physiology. So we are doing this chapter. So we are done with the first part of the chapter. So now we are in the second half of this chapter. So join with me to see how your urine formation happens. So there are 3 steps which we are about to talk about. So let's talk about that. And we'll also talk about the function of each of the tubules. Yes, so thank you so much for joining my session. So please do like, share and subscribe. So come on, share with your friends. So let your friends know that here ma'am is teaching you, because everyone needs guidance at one point and everyone needs help at one point, right? So share it with your friends so that they will also gain the benefit of this class. Yes, come on. So please do like, share and subscribe. So make it as quick as possible. So quickly we'll enter into the subject. Yes. So I hope all those who are appearing for NEET 2021, so you are preparing really hard, so you're getting the concepts done, you're preparing, you're revising the concepts, you're solving enough questions. Yes, can I believe that? So we are nearing the exam date. So how much is your countdown right now? So today we are in 20th of July, so you have 11 more days in July. So the entire of August and 10 days there. So you almost have 51, so almost you have 54 days, right, to be precise. So not more than that. Yes, Shimpi, hello, a very happy good evening. I'm good, thank you. How are you? So thank you, Shimpi, for joining my lecture. So please do like, share and subscribe. So how is Munna? Okay, so shall we talk about excretory substances and their elimination? Okay, so we have been talking about the description of your kidney, your structure of your nephrons. So today I will give you a quick recap and then we'll go forward with the mechanism of urine formation. Alright, yes, so here we go. So let others join in as and when we move forward. So you guys have to send me suggestions on what type of sessions you want for 2021. So Satyendra told mock test series, so that is taken. So others, please type in your suggestions in the comment box, not in the live chat. Alright, yes, Anthony B, yes, welcome to my session. A very happy good evening. So please do like, share and subscribe. Okay, so here we go. So excretory substances and their elimination. So this is what we are going to talk about. So we have to talk about the human excretory system, which we started. So today we will talk about the process of urine formation, the functions of tubule. If time permits, we will see the introduction for the mechanism of concentration of filtrate, right? So in the next class, we will wind it up. So talking about the regulation of kidney function, micturition, and finally the disorders of your excretory system. So with this, the chapter ends. Hi Munna, yes, hello, how are you? So thank you so much for joining my lecture. So please do like, share and subscribe. Yes, I'm fine, thank you. Okay, so what are the parts of your human excretory system? So we were talking about the human excretory system. So we will start from there. So human excretory system consists of a pair of kidneys, so we have a pair of ureter, so we have one urinary bladder and we have urethra, right? So these 4 things make up the excretory system. Okay, so you are good, fine. So you and Shimpi are not fighting these days, things are fine between you, yes? Okay, so now then we went on to talk about the structure of the kidney. So what is the structure of the kidney? So kidney is reddish brown in color, it is bean shaped and it is located between the last thoracic and the 3rd lumbar vertebrae, and it is attached to the inner dorsal abdominal wall, right? So that is the location of your kidney. So we also spoke about the dimensions of the kidney, which is not a very precise that you have to remember. So but anyways, so it is from 10 to 12 centimeter. So then it is 5 to 7 centimeter in length and thickness is 2 to 3 centimeter, right? So now talking about the kidney, so on the concave surface of the kidney you have a notch. So the notch of the kidney is known as hilum. So on the concave side you have a notch that is known as hilum. So this is where your nerves, so your blood your ureter, everything enters the kidney, right? So inner to the hilum, okay, so just inner to the hilum, you have what is known as your renal pelvis. So inner to the hilum, so this is inner to the hilum. So you have funnel shaped structures which is known as renal pelvis. So it has projections, so which is known as calyces, right? So which is known as calyces. Till this, I hope you are able to recollect. So this is how you should recollect. So this is known as calyces. The projections on your renal pelvis is calyces. So Shimpi is the criminal here. Is that so? But Shimpi is also a good person. So don't worry. Next time if something like that happens and if she does not score marks, so come and tell me. All right. Yes. Hello Leela. So good evening. So thank you for joining my lecture. So please do like, share and subscribe. So Glorious. Yes Mary. Hello. Good evening. So thank you for joining my session. So please do like, share and subscribe. So Shimpi, you are being the bullying elder sister, so this is not acceptable. So Munna is a kid, so don't do like this with her, okay? Yes, done. So you have renal pelvis which has projections known as calyces, right? So the kidney is covered by an outer tough capsule. So this is on the outer side. So on the inner side, the kidney is divided into two parts. One is the outer medulla. The outer cortex and the inner medulla. So the medulla opens into conical masses, so which is known as medullary pyramids, which projects through these calyxes. Right? So your cortex region flows through these medullary pyramids as renal columns, which is known as columns of Bertini. Which is known as columns of Bertini. All right? Yes. Okay, you are Anthony. Okay, fine. Okay, Anthony, you can message from this only. All right, that's fine. Okay, no issues. Okay, so this is known as columns of Bertini, right? So now, next we started talking about the structure of your nephron. So we have nearly 1 million structures. So we have nearly 1 million nephron. So this is the functional unit of your kidney. Right? So the nephron has three structures, okay? Sorry, it has two structures. One is your glomerulus and then it has your renal tubules. So we have two parts of your nephrons, okay? So what is glomerulus? So glomerulus is a network of capillaries. So it is a tuft of capillaries. So tuft is a network of capillaries. Alright. So here we have two capillaries. So one is afferent arteriole and the other one is efferent arteriole. Right? So this brings blood into the glomerulus and this takes blood away from the glomerulus. So afferent brings in, efferent takes out. Okay? Fine. So this is a branch of your renal artery. So artery carries oxygenated blood. Right? So this is your glomerulus. Fine? Right. So now we are going to talk about the renal tubule. So the renal tubule extends from a cup-shaped structure. You have a cup-shaped structure like this. So this is your glomerulus and this is your Bowman's capsule. So together it is known as your Malpighian corpuscles. It is your Malpighian tubule, or it is known as your renal corpuscles. Right? From here, your renal tubules begin. So now this continues to form a highly convoluted structure. So this is known as your PCT, proximal convoluted tubule. Okay? So then you have an hairpin shaped structure. So this is known as the loop of Henle. Right? So this has your descending lymph and this has your ascending lymph. So again you have a lightly coiled structure. So this is your distal convoluted tubule and then it opens into the collecting duct. So this is the collecting duct. Alright. So this is loop of Henle. So this is the ascending and this is the descending lymph. Right. So I hope the structure of nephron is clear. So the collecting duct opens into the renal pelvis through renal medulla or through medullary pyramids. Alright, so this is the structure of your nephrons. Alright, okay Antonia B, that's really great. Yes Naveen, hello, welcome to my lecture. So thank you so much for joining my lecture. So please do like, share and subscribe. Okay. Oh, Munna is lying. Oh, how much angry Munna is getting? Okay. So now I cannot believe either of you. I have to talk to your mom. Only your mom knows the whole story, right? So I don't know who's lying and who's telling me the truth here. Okay, one day I will have parents teachers meeting. So I will talk to your mom. Okay, done. So this is about the structure of the nephron. So we went further talking about what is cortical nephron and what are juxtamedullary nephrons. So if you see your Malpighian tubule, so that is here. So your Malpighian tubule, your PCT, your DCT, so everything remains in the cortex region of your kidney, right? So sometimes loop of Henle also belongs to the cortex region if it is too small. So if loop of Henle is also present in the cortex region, then it is known as cortical nephrons. So it is known as cortical nephrons. Alright. So if the loop of Henle is long enough and it extends into the medullary region, so it is known as juxtamedullary nephrons. Right? Okay. So now moving further, your efferent arteriole. So this is your efferent arteriole and this is your efferent arteriole. So this efferent arteriole forms a network of tubules that is known as peritubular capillaries around your renal tubules. So your efferent arteriole, so I will write it here. So your efferent arteriole, so this forms a network of, okay, so it forms a network of capillaries around your renal tubules, which is known as peritubular capillaries. So among these capillaries is a minute U-shaped blood vessel, so that runs parallel to your loop of Henle. You have an U-shaped blood vessel. So this runs parallel to your loop of Henle. So this is known as vasa recta. So vasa recta and loop of Henle are opposite in action. So if this is my loop of Henle, so this is the hairpin shaped structure. So this is my descending lymph and this is my ascending lymph. this is my loop of Henle. So now my vasa recta is like this. So in my vasa recta, suppose if this is my vasa recta, so here this is the ascending limb and this is the descending limb, okay. So this is vasa recta, right. So this is what we spoke in the previous lecture. So then we went forward to talk about the mechanism or the formation of urine. So for this we have three main steps, right. So here you have certain pressures also which you have to remember. So pay attention, I will give you an outline of what are the different pressures that come here, okay. So urine formation involves three steps. The first step is your glomerular filtration. The second one is tubular reabsorption. Then you have tubular secretion. Fine. So now let's talk about your glomerular filtration. So we are talking about this. So I will explain this once again. So how does your glomerular filtration happen? So here what is involved? Your glomerulus, right? So we are talking about glomerular filtration. So filtration done by the glomerulus is your glomerular filtration. So glomerulus, where it is present, it is a network of capillaries. So you have this structure, right? So this is your glomerulus. So here you have a capillary. So this is your afferent arteriole. So here you have another one. So this is your afferent arteriole. Fine. So this is your Bowman's capsule, which we know, right? So together it is known as the Malpighian tubule or the renal corpuscle. Now, if I say filtration is going to be done, so what is going to get filtered? So blood is going to get filtered. So blood is going to get filtered, alright. So for the process of filtration, so who are participating? So the participants in this process of filtration is the blood vessel of your glomerulus. So glomerulus is a network of blood capillaries. So the blood capillaries, which is known as endothelium, okay. So the first layer is the endothelium of the glomerulus. Okay. Then you have epithelial cells in your Bowman's capsule. So epithelial cells of Bowman's capsule. So epithelial cells of Bowman's capsule has a special name which is known as your podocytes. Fine. And the third one, so in between these two, we have a basement membrane, right? So the basement membrane between these two. So these are the three layers which are going to get involved in your glomerular filtration, right? So your blood is going to get filtered here. So this filtration is so accurate because your podocytes have what is known as filtration slits or filtration pores. So they filter tea, right? So when they filter tea, you can see minute holes. So it will be much smaller than these. The purpose of having podocytes is because they have filtration slits. They do not even allow plasma proteins to get filtered. So even plasma proteins are retained. So that is the function of your podocyte. So since the filtration is very accurate, so this filtration is known as ultrafiltration. Okay. So, how much blood does it filter? So, the amount of blood that is filtered by your glomerulus. So, blood is filtered by your glomerulus, right? So, this blood that is filtered by your glomerulus is known as your glomerular filtration rate. So, the value of these is 125 ml per minute. So, you have 125 ml of filtrate per minute. So, in a day, you have 180 liters per day. So, this is the amount of blood that it filters in a day. Alright? Yes? Okay. So, where does this blood come from? So, we know that ventricles of the heart, each ventricle pumps about so much of blood, okay? So, around 1100 to 1200 ml. So, this accounts to 20 to 30% of the blood, right? So, that enters into your kidney. The amount of blood that enters inside your kidney is approximately 650 ml of your blood plasma. So, this is the total blood, but we only need the plasma. So, the blood plasma is 650 ml which enters the kidney for filtration. So, this is known as renal plasma flow. So, RPF, the blood plasma that enters the kidney is your renal plasma flow. Okay. So, this is your GFR, right. So, I hope this much is clear. Yes. So, now shall I move forward? Okay. So, now moving further. Okay. So, here we talk about absorption and okay. So, before we go to that, so we have three types of pressures which is very important. So, the filtration that you have, that is actually accounted by three types of pressures. Yes ma'am, good evening and welcome to my lecture. So, please do like, share and subscribe. Okay. So, we have three types of pressure which determine the effective filtration process. So, if the filtration has been done properly or not, so this is overlooked by three pressures. The first pressure is your glomerular hydrostatic pressure. It is your glomerular hydrostatic pressure. So, this is in short referred as GHP. So, this is the pressure in the capillaries of your glomerulus. So, this is the pressure in the capillaries of the glomerulus. So, among the three pressure that we are going to talk about, so this is the chief determinant. So, this pressure is very, very important. So, this pressure arises due to the difference in diameter of your afferent and your efferent arteriole. So, that is why you have this pressure. So, the value of your GHP is around 60 to 70 mmHg. Okay, it is around 60 to 70 mmHg. Fine. So, the second type of pressure is your colloidal osmotic pressure. So, this is known as COP. Okay. So, colloidal osmotic pressure is the osmotic pressure created in the blood. So, because of this osmotic pressure only, your blood plasma or the plasma protein is not filtered. Alright. So, here the value of COP is almost 30 or to 20 mmHg. right? So this is the second type of pressure, that is colloidal osmotic pressure. Yes, Ayappan Shaktivel, good evening. Yes, I am good, thank you. How are you? How are you? Okay, fine. The third type of pressure is your capsular hydrostatic pressure. So this is the last pressure. So what is capsular hydrostatic pressure? So this is the pressure that is covered by the fluid that is coming from the Bowman's capsule. So pressure which is present in the fluid of the Bowman's capsule. So the Bowman's capsule filters, right? The pressure in that filtrate or the fluid is your capsular hydrostatic pressure. So the value is 10 to 18 mmHg. Right? So this is the value of CHP. So effective filtration pressure is equal to, so glomerular hydrostatic pressure minus COP plus CHP. Okay, so here we take this value as 60. So then we move forward. So we take this as 32 plus 18, so which is 50. So the net effective filtration pressure is 10 mmHg. So we take the average values. Is this clear? Yes. If you start revising now, then you can get a good score. But if you starting from scratch, then yes, it is not going to be a good path. So you have to struggle really hard. Yes, Dharini, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. For all those who joined just now, I am talking about glomerular filtration. So glomerular filtration. So if you want to know if your filtration is effective or not, so that is determined by 3 factors. Okay. So the 3 factors is your glomerular hydrostatic pressure, colloidal osmotic pressure, and then your capsular hydrostatic pressure. Alright, okay. So glomerular hydrostatic pressure is the pressure in the capillaries of the glomerulus. So it is due to the difference in the diameter of your afferent and your efferent capsule. Okay, yes. So here the value is 60 to 70 mmHg. So the next one is colloidal osmotic pressure. So that is the osmotic pressure created in the blood vessels. So that is why you do not have the plasma proteins getting filtered. So here the value is 30 to 20 mmHg. Then the last one is capsular hydrostatic pressure. You have the filtrate coming out of Bowman's capsule, right? The glomerular filtrate. The pressure in the glomerular filtrate is your capsular hydrostatic pressure. The value of this is 10 to 18 mmHg. So the effective filtration pressure is equal to, so GHP minus the sum of these two. So I have taken the average values. So the effective filtration pressure is 10 mmHg. Alright, done? Okay, so this is about the pressure that I want to tell you. So now we move forward. So I want to tell you, okay, so this is, I told you what is glomerular filtration pressure. Okay, so suppose if your glomerular filtration pressure falls down. Oh my God, we have come towards the end. Okay, so suppose if your glomerular filtration pressure falls down, so then what will happen? So how is the regulation? So you have a particular GFR rate, right? So this is 125 ml per minute or 180 liters per day. But if this falls down, this glomerular filtration rate falls down and thus the glomerular filtration pressure decreases, then how will you regulate it? So this is regulated by a specialized type of cell known as JGA cells. So JGA stands for juxtamedullary apparatus. So juxtamedullary sorry, it is juxtaglomerular apparatus. I am sorry. So it is juxtaglomerular apparatus. Okay, so these cells are present in the junction between your DCT and your afferent arteriole. So afferent arteriole is the one that brings blood into the glomerulus. So DCT is distal convoluted tubule, right? So in the junction between these two, you have juxtaglomerular apparatus cells. So these cells produce a particular type of enzyme which is known as renin. Okay, so this renin enters your bloodstream and it does some activation. So what is the activation? So this renin converts angiotensinogen into angiotensin II. So now if your angiotensin II is activated, so this causes constriction of the blood vessel. So here your pressure is low, right? So which means the volume is high. So volume and pressure are inversely related. Now if I want to increase the glomerular pressure, I have to decrease the volume. So I am constricting the blood vessel so that my glomerular pressure increases. So this is how the regulation happens. Is this clear to everyone? How the regulation happens? Yes, no doubts. So this is how the glomerular filtration rate is regulated within your kidney itself. Done? Okay. So we know the glomerular filtration rate is 125 ml per minute or 180 liters per day. Suppose if your glomerular filtration rate falls less, if it is 110 ml per minute. Okay, so if it falls below the normal value and thus the glomerular pressure also decreases, so how will you bring it back to normal? So you have a group of cells known as JEA cells. So JEA stands for juxtaglomerular apparatus. Okay, so these cells are present in the junction between your distal convoluted tubule and your afferent arteriole. Alright. So these cells, they release a particular enzyme known as renin. So renin. So renin is in your digestive system. Okay. So renin is secreted by your JG cells into the bloodstream. So this renin converts angiotensinogen into angiotensin II. So this angiotensin causes constriction of the blood vessels. Why? So if I say the pressure is less, then the volume of the blood vessel is greater, right? Because pressure and volume are inversely proportional. Now if I want to increase the pressure, I have to decrease the volume. Thus your angiotensinogen constricts, that is reduces the volume of your blood vessel. So this is how the regulation happens. So Shimpi, did you understand how the regulation happens? Yes, any further doubts? Any further doubts, or is this clear to everyone? Is this clear? Shimpi? Done? Yes, okay. So this is about glomerular filtration. Now the next step is reabsorption and secretion. So the next step, reabsorption. So let's talk about reabsorption. So if you see, we have 180 liters per day of the filtrate which is filtered by your kidney, right? Right. So this is how much your kidney filters. But how much urine do you pass? So in a day, we pass only 1.5 liters of urine. So 1.5 liters, but here we have 180 liters. So almost 99% where it goes then. So 99% is getting reabsorbed by your renal tubules. Okay, so this is the next process, that is reabsorption. Alright, yes Mary, good evening. So welcome to my session. So sorry for getting back late to you. Please do like, share and subscribe. Alright, okay. So this is reabsorbed by your renal tubules, that is your DCT, PCT and the collecting duct, either actively or passively. Done? Okay. So what is getting reabsorbed? So first let's talk about that when you're talking about the function of the tubules. Okay. So you have reabsorption of your Na+, K+, glucose, amino acids. So all these are reabsorbed actively. So water is reabsorbed by the process of diffusion. Now there was someone else known as Mercy Mary, so I wished them. Okay, yes, you I know, this is Anthony, okay, right? So this is all reabsorbed actively. The next step in the process of urine formation is secretion. So we are done with reabsorption. So this is reabsorbing all the necessary items. Now it is secretion. Okay. So to maintain ionic balance of the body fluids, certain ions are secreted into the filtrate. So glomerulus filters the blood. Yes. So in that blood, you will have a loss of ionic balance because your blood plasma has electrolytes as well, right? So when you filter and when it is getting reabsorbed, so there might be a loss of the osmotic balance. So to maintain the pH once again, certain ions are secreted into the filtrate. So this is also done by your renal tubules, alright? So you secrete H+, K+, ammonia into the filtrate to maintain the balance, right? So this is what happens. So this is the third step of tubular secretion. So we spoke about reabsorption and then we have tubular secretion. Alright, yes, filtrate, sorry. Done? Is this clear? So the mechanism of urine formation, are we done? Yes, thoroughly? So this I have explained how the JEE cells regulate themselves. So yes, reabsorption and secretion is also done. So now shall we talk about the function of tubules? So the function of the three tubules that we have. So we have four tubules. What are the tubules we have? Proximal convoluted tubule, Henle's loop, distal convoluted tubule, collecting duct, right? So we will talk about the function of each of them. So that's the next topic, that is mechanism of concentration of urine becomes easier. So let's see how it happens, okay? So let's talk about your PCT. So this is your proximal convoluted tubule and it is a bit highly coiled, right? So this is lined by brush border epithelial cells. So this is lined by brush border epithelial cells. Okay. So this is a type of renal tubule and renal tubules are involved in reabsorption and secretion, right? So maximum reabsorption happens here. So maximum reabsorption happens in your PCT. So all the essential nutrients and the electrolytes are reabsorbed here. Okay. So like your Na+, K+, so all the important electrolytes are reabsorbed here. So this plays a role in reabsorption as well as in secretion. So it also secretes H+, K+ and ammonia into the filtrate. So this is the function of your PCT. So PCT is easy to study. Okay. So now we go into the, yes. So almost 70% is reabsorbed here. So this includes your essential nutrients and your electrolytes. Yes, you're absolutely right. Okay. So now we are coming to the most tricky Henle's loop. So what does Henle's loop have to say? So Henle's loop has two parts, right? So what are the two parts of the Henle's loop? So first we have the descending lymph, and then we have the ascending lymph, right? It's not lymph, it's lymph. Descending lymph and the ascending lymph, right? So the descending, so it is like this, and then it becomes like this. So this is how your exact structure is, right? So this is my descending lymph. So here it is made up of columnar epithelium. So here it is made up of squamous epithelium. So here it is made up of columnar epithelium, and here it is squamous epithelium, right? So your descending lymph of Henle is permeable to water and it is impermeable to electrolytes. So what do I mean by permeable to water? Which means water can move in and out of the walls of your descending lymph, right? Yes. So here your filtrate comes from your Bowman's capsule. So reabsorption happens, and here since it is permeable to water, so water moves out of the filtrate. So filtrate is passing inside it. So since the walls of this is permeable to water, so water moves out of your PCT. Thus the filtrate is concentrated. So here your filtrate is concentrated. Okay, right? So the next part of your Henle's loop is your descending lymph. So this is like this. So this is also similar to this, but my figure is wrong. So this is climbing up. So this is also lined by columnar epithelium, and this is by squamous epithelium. But the ascending lymph of your Henle's loop is permeable to electrolytes but it is impermeable to water. So now when the fluid is coming like this and moving into the ascending lymph, the electrolyte moves out from here. So what remains? Only the remaining fluid, that is water. So here the filtrate is diluted. Is this clear? So why the filtrate is concentrated here and why it is diluted here is clear to everyone? Yes? Any doubts? Ask me right now. Quick. Is this clear to everyone? No doubts? Done? So can I move forward? So Naveen, Shimpi, Anthony. So Leela, did you understand? Yes. Okay, fine. So I am moving further. So now we are talking about your DCT. Okay. So what is the role of, okay, so I have the picture. So this is the columnar epithelium. Okay. So if you just, okay, so this is your columnar epithelium, the figure that comes, right. So moving further, so now, okay, so here I had space for PCT, but I wrote there itself. Okay, alright. So now let's talk about your DCT. So distal convoluted tubule. So what happens here? So here you also have reabsorption happening. But here the reabsorption is conditional. So based on some conditions, so you have reabsorption here. So here you have the reabsorption of Na plus and water to some extent. Okay. So this also has permission to reabsorb urea. So what causes conditional reabsorption of your DCT? So you have a hormone which is known as aldosterone. Yes, so this aldosterone is produced by the adrenal cortex, okay. So it is a hormone of your adrenal gland. So adrenal cortex produces mineralocorticoids. One among them is aldosterone. So this causes the reabsorption of Na plus and water. So if your aldosterone is not there, right, so then it becomes difficult because that helps in reabsorption. So this is your function of your DCT, right. The next one is the function of your collecting duct. So the last one, so collecting duct opens into renal pelvis, right, through your medullary pyramids. So now collecting duct reabsorbs water and urea to a larger extent. So this is the function of your collecting duct. Apart from this, your DCT helps in conditional reabsorption. It also helps in tubular secretion. So collecting duct also helps in tubular secretion. So this is the function of each of these tubules. Is this clear? Yes. So today I have finished the formation of urine and the function of each of these tubules. Done? Yes. So with this I will stop. So in the next class I will teach you the mechanism of concentration of filtrate, and then we will talk about the disorders and regulation. So we will be done with this chapter maximum by this week. So tomorrow at 5 p.m. we are winding up evolution. So you have a Menti quiz on Thursday. So yes, I will plan up something new because for all those who are preparing for 2021, yes, I will bring a one-shot revision series so that it is useful for all of you. Maybe at the max we will discuss only questions. Okay? Done? So please do type in your suggestions in the comment box, and tomorrow at 5 p.m. we are finishing off evolution. So we will be talking about the mechanism of evolution, Hardy-Weinberg principle and evolution of man. So join me for the same, alright. So if you genuinely understood my way of teaching, so please do like, share and subscribe, and stay tuned. I will meet you tomorrow. So until then, this is me signing off. Take care and bye.